Very good afternoon, very good evening, everyone. I'm Dr. Chaitali Nikam from Thyrocare Technologies Limited. I'm heading infectious diseases section at Thyrocare Technologies, named as a Focus TB. We are having RNTCP, NABL, and CAP accredited labs since 4 years, and with the help of or in collaboration with Asst Analytics, we have recently launched a new test for whole genome sequencing for a TB in very less price as compared to market, and we are conducting this webinar today along with Dr. Nikhil Sarangdhar. So before introduction to Nikhil Sarangdhar, I just want to suggest few of the comments from our end, that after Dr. Nikhil's presentation, there will be a question and answer session. If anyone wants to ask any question, they can write it in the chat box, and after the session, Dr. Nikhil will take it one by one. Also, if anyone wants to ask the question directly to anyone of the panelists or the moderator, they can raise their hand. So I'm just giving the brief introduction of Dr. Nikhil, as he has already presented once, and at that time the chat box was flooded with lots of positive vibes, and for the best presentation which we have got on this platform. So Dr. Nikhil Sarangdhar is a chest consultant with independent practice in Mumbai. He's a former assistant professor at Department of TB and Chest Disease at KJ Somaiya Medical College, Mumbai. He is organizing secretary of NAPCON at 2016 and on 2020. He's organizing secretary of 16 national conferences and 9 workshops. And he's a recipient of 8 prestigious Young Scientist Award. Research list is too long, yeah. He's recipient of NCCPI for P.S. Shankar and Professor K.C. Mohanty Chest Oration 2020. He's editor of NCCPI newsletter, that is Lung Bulletin, and author 27 chapters in textbooks and 18 articles in general. So basically, I'm giving warm welcome to our chief guest speaker, Dr. Nikhil Sarangdhar, and over to you, Dr. Nikhil.
Thank you, Dr. Chaitali, and all panelists from Asst Analysis and Thyrocare. So you can proceed, then I can begin my presentation.
Yes, Dr. Nikhil, please share the presentation.
Okay.
Please share slides, Ranjit.
Yeah.
Dr. Nikhil will be presenting the slide actually.
Yeah.
Yeah.
Just a moment. Just a moment. Yeah. Screen visible, slides visible?
Yes,
Yes, slides are visible. You need to just make it full screen. So.
Yeah, I'll do that. Just a moment. Yeah, can I begin?
Yes, sir.
Yeah. So good evening, everybody. At the outset, I'd like to thank Thyrocare and Asst Analysis for inviting us to be here. My topic is early diagnosis and DST with TB whole genome sequencing. Can it mitigate the existing challenges of management of multi and extensively drug-resistant TB? So before I begin, I'll pay a tribute to the departed soul of my late teacher, who was often given the award of the Bhishma Pitama in tuberculosis at the hands of the President of India, Shri Pranab Mukherjee in 2014, late Emeritus Director Professor Dr. K.C. Mohanty. So, colleagues, what is the problem with existing facilities for MNXDR TB in India? First thing is there is, of course, a delay in treatment, but before that, there is always a delay in diagnosis. So when it comes to what is the first thing I would like, I would say the first thing is we should have a rapid diagnosis. To illustrate this, you have, instant coffee on the left and you have filter coffee on the right. But what I'm trying to say is that some things we need, so we have to take shortcuts, but sometimes those shortcuts may not always be good. There are always advantages and disadvantages, pros and cons of everything. So let us discuss what these are in short. So now you see the first thing for better management of MNXDR TB is rapid diagnosis. So when it comes to TB diagnosis, we have clinical diagnosis when the patient comes with signs and symptoms, radiological diagnosis when you image the lungs by an X-ray or an extrapulmonary TB, the affected site by ultrasound or CT or other imaging modalities. You have pathological diagnosis, that is a tissue diagnosis by invasive methods, and you have microbiological diagnosis. It could be of sputum, it could be of other isolates. Clinical and radiological diagnosis are subjective diagnosis, and pathological diagnosis are definitive diagnosis. Right. If you ask me what is the most rapid test to diagnose TB amongst these, you have in the beginning sputum microscopy for acid pass bacilli. You have in the middle gene expert, that is your cartridge based NAT, and you have last but not least the chest X-ray. So if you ask me for pulmonary tuberculosis, the chest X-ray is still the most rapid method to diagnose TB. Nowadays, X-rays are digital and you get the film in front of you in a few minutes. For decades, even prior to the advent of NTEP or RNTCP as it was called, it was the basis for TB diagnosis and treatment. Chest X-ray still remains the first investigation of choice in pulmonary or pleural disease. And even in patients with extrapulmonary TB, a chest X-ray should be taken to look for any coexisting pulmonary pathology. So when it comes to diagnostic tests for TB, you have two types of tests: direct tests, which detect the presence of Mycobacterium tuberculosis in the body, and indirect tests, which assess the body's reaction to TB infection or disease. So amongst the tests we have in hand for diagnosis of TB, we have microscopy, Bio-Nielsen and modified acid fast stains. You have fluorescent microscopy. You have culture with drug susceptibility testing, that is your phenotypic culture, which can be solid or liquid. You have molecular techniques for genotypic tests like Gene Expert, line probe assay, and others which we will be discussing shortly. So just to brief you about it, culture is of two types, solid and liquid. It remains the gold standard. It has several advantages. It fulfills Koch's postulates for a definitive diagnosis of Mycobacterium tuberculosis infection. It provides specific standardized drug susceptibility testing, rendering individualized treatment possible. It differentiates Mycobacterium tuberculosis from non-tubercular mycobacteria. It's highly sensitive and specific to the order of 10 bacilli per ml, even for extrapulmonary samples. And you get culture conversion with successful therapy, so it can be used as a monitoring tool. The disadvantages is the time. You see, this is the gold standard, and a solid culture takes anywhere from 6 to 8 weeks, and liquid cultures also 2 to 3 weeks minimum. Cultures are expensive. Culture media are prone to contamination, and you have biosafety hazards of aerosol generation, particularly in these times of COVID with your liquid cultures. So now to have some of the advantages of rapid diagnosis to retain sensitivity and specificity for TB diagnosis and do away with some of the drawbacks of culture. In the last 2 decades, we have seen the advent of genotypic tests, or molecular diagnosis as we call this. What do these rapid molecular tests do? They detect the presence of Mycobacterium tuberculosis in the specimen. They identify the mycobacterial species as Mycobacterium tuberculosis, and they also identify drug resistance to key anti-TB drugs. So the first of these to be rolled out on a large scale was the gene expert or cartridge-based nucleic acid amplification test, CBNAT it is also called. It was rolled out in the first decade of the century by the WHO. It's a very simple test. You have sputum liquefaction and inactivation with the reagent, transfer 2 ml of this into the cartridge, and then insert it into a gene expert machine. There your hands-on work is over. So what it does, it processes the sample. There's ultrasonic lysis of the organisms in the sample to release the DNA. This DNA is amplified with a PCR reaction, with PCR reagents. So it's a polymerase chain reaction. And then finally you get the test report as whether MTB is detected or not. If it is detected, it's quantified as low, very low, medium or high. And if detected, whether rifampicin resistance is there or not. The time to result is 1 hour and 45 minutes, but actually it's not so simple because it takes almost a day for the report to come in hand from the time you send the sample. Keep in mind that gene expert will only identify RIF resistance and not MDR-TB per se. But we take RIF resistance as a surrogate marker for MDR-TB because we know that patients whose culture isolates have Mycobacterium tuberculosis with RIF resistance will fail a first-line regimen or a rifampicin containing regimen. So they have to be started on a treatment regimen for MDR-TB. Coming to the line probe assay, this also identifies the MTB complex. It detects mutations associated with resistance to INH and rifampicin, that is the genotype MTBR plus or enolipar TB. This gives you a diagnosis of MDR TB. And the second line, LPA or the genotype MTBRSL, it detects resistance to ofloxacin, amikacin, and ethambutol. And this will give you a diagnosis also of pre-XDR or XDR TB. So LPA has good sensitivity and specificity, but only when culture isolates are used. So you see, if you compare the sensitivity and of existing tests for TB, at the bottom of your diagnostic pyramid is the midget, that is your liquid culture, where you require only 10 to 100 ml of bacilli, 10 to 100 bacilli per ml of sample. Then you have your CBNAT, which is your gene expert, which requires about 150 bacilli per ml, and then you have your line probe assay, which requires at least 10,000 bacilli per ml, equivalent to that of your sputum. So coming to the advantages of gene expert, as I told you, it gives you rapid results. It helps you enroll more RIF resistance patients on MDR-TB treatment faster. It quantifies your MTB load as very low, low, intermediate, or high. It has the least biohazard risk of all the tests, and there is good sensitivity for both smear negative, smear positive, as well as extrapulmonary samples. The disadvantages of gene expert are like all the genotypic tests. It remains positive even in patients who have been cured and completed treatment. Therefore, it cannot be used as a monitoring tool. And the diagnostic accuracy is limited, you see, because it depends on the cycle threshold. So if the sample is paucibacillary, that is to say your MTB load is low or very low, then sometimes you may have a problem interpreting the test. So you see, even for extrapulmonary samples, CBNAT or gene expert has been compared with culture. And though some studies have found good results, others have found discordant results also. Coming to your line probe assay, the advantages are it again gives you rapid results. It helps, apart from giving you a diagnosis of MNXDR TB, it will also help you enroll patients on treatment faster. There is potential for improved yield of results relative to culture positivity and less biohazard risk if there is no culture manipulation, somewhere between that of gene expert and liquid culture. The disadvantages are that there is risk of DNA contamination, cross contamination. You see, the test may fail if inhibitors of DNA or PCR inhibitors like blood are present in the sample, and there is a limited number of drugs for which the assay is standardized. For example, it will only give you INH, rifampicin, a quinolone, and an aminoglycoside and ethambutol. But there are at least 18 to 19 anti-TB drugs in the pipeline which we use while designing a regimen. It will not give you resistance or sensitivity pattern to the other drugs. So all said and done, The molecular methods are good. They give you a rapid diagnosis, but they still have their limitations. We've discussed they have variable sensitivity in extrapulmonary disease, smear negative pulmonary disease, TB HIV co-infection, and when dealing with samples from children affected with tuberculosis. So therefore, while they do overcome some of the drawbacks of culture, they cannot be recommended to replace conventional culture methods entirely. So this is how we go when you have a culture positive for tuberculosis. You go by your conventional testing, biochemical tests, antigen detection, and then bio growth characteristics and drug susceptibility testing, for which you can further analyze by molecular methods. It is important to identify the isolate as Mycobacterium tuberculosis complex before DST, because NTM or non-tubercular mycobacteria can be resistant to your conventional anti-TB drugs, and therefore doing a DST without speciation can result in inappropriate therapy. This is what the diagnostic pyramid, this is what the diagnostic algorithm for tuberculosis is. This is for pulmonary tuberculosis. Now, if you see you have a patient with clinical symptoms suggestive of TB and chest X-ray abnormalities suggestive of TB. So initially, we used to go for a sputum smear based on whether the smear was positive or negative. We used to go for a CB NAT. Then, depending on that, whether the NAT was positive or not, you would consider it a confirmed case of TB. If it was negative, you would go for culture. Nowadays, you go for first and second line LPA. So you see, this whole process takes a lot of time. It's not a simple thing, and any delay in diagnosis will lead to a delay in treatment. I'll come to that later in my talk. And these were the original standards for TB care, you see, which were in 2014, which have been revised now. So you see, they used to rely on a microbiological confirmation on sputum, then chest X-ray as a screening tool, then CB NAT or your gene expert, etc., etc. And for patients with extrapulmonary TB, again, they said that clinical specimens must be taken from the sites of involvement for testing for microscopy, culture, DST, CB NAT, molecular tests, or histopath examination. So this whole process used to take time. Now we have over there certain advantages in the rapid molecular methods, and we'll go through those one by one. You have the Gene Expert Ultra, which has a larger chamber for DNA amplification, enhanced sensitivity, 16 bacilli per ml of sputum as compared with the original 130 per ml for Gene Expert. 2 additional molecular targets which give you better sensitivity. There's a less likelihood of false positives, but they are still there. And you can use the Expert Ultra cartridge along with your existing Gene Expert equipment. All you have to do is upgrade the software. Then you have TrueNat, which is an indigenous CB NAT from our country, which requires only half an ml of sputum for testing as compared with your 1 to 2 ml for Gene Expert. It's battery operated and portable. It has good sensitivity and specificity, and you get results still faster. So what are the advantages in culture methods? You have microscopically observed drug susceptibility, or the MODS assay, the nitrate reduction assay, or the NRA, or the colorimetric redox indicator, or the CRI. Some of these, like the MODS and NRA, can be done directly on sputum, and all the three can be done indirectly on isolates. So coming to the MODS, it's on liquid media, increased sensitivity and a faster growth, faster turnabout time. Standardized assay and testing protocol is available. It has very good sensitivity, and the cost of consumables is very inexpensive, just about $2. Then your NRA, or the nitrate reduction assay, is based on the ability of Mycobacterium tuberculosis to reduce nitrate to nitrite, seen as a color change of LJ media using grease reagent versus of solid culture. It is sensitive, it improves the turnabout time, and is also inexpensive. In your colorimetric redox indicator is based on reduction of an indicator by metabolically active MTB. It is indirectly applied from isolates of from LJ or liquid media. You have a MIC determination with microdilution plates. In 7 days, you incubate the plate, add the indicator, and then you get a color change and then growth of MTB. Coming to what we are to discuss today, TB whole genome sequencing. What does it mean? So it's also called full genome sequencing, complete genome sequencing, or entire genome sequencing. This is the process of determining the entirety or nearly the entirety of the DNA sequence of the genome of any organism at a single point in time. So how is it applicable for TB diagnosis? Friends, resistance in Mycobacterium tuberculosis is conferred mainly by nucleotide variations. And what whole genome sequencing does is use high resolution sequencing technology to interrogate the entire TB genome to identify drug resistance across all the anti-TB drugs that are used. It will also be helpful in identifying cases of mixed infection, co-resistance, co-infection, or heteroresistance. Whole genome sequencing is now being increasingly recognized and adopted for the diagnosis of DR-TB and treatment in many parts of the world. So this is how it is. You collect the sample, enrich by culture, DNA extraction, then pre-sequencing preparation, sequencing and data analysis. That is the Omega TB test, and then the generation of report. So you see you have 18 drugs here to which whole genome sequencing can be done. They have been tested mainly on pulmonary samples, and I think there is research going on for extra pulmonary samples also. So the same drugs that you test on culture, you are able to test on whole genome sequencing, which is an added advantage over existing methods like GeneXpert or line probe assay. And what are the performance metrics of whole genome sequencing? What is the sensitivity and specificity? As you can see here in this slide, sensitivity and specificity are above 90% for all the first line anti-TB drugs, and at least above 85% for most of the second line anti-TB drugs also. And then when you compare with the gold standard, that is culture, if you compare GeneXpert, the agreement between culture DST and GeneXpert is just 49%. With LPA, it's 63%. With whole genome sequencing, it's 93%. I think that's wonderful. So you see whole genome or next generation sequencing has great potential as a method for rapidly diagnosing drug-resistant, multi or extensively drug-resistant TB in diverse clinical reference laboratory settings worldwide. And it overcomes many of the limitations that are associated with your cultures or with less comprehensive genotypic methods like GeneXpert and LPA by giving you detailed sequence information for multiple gene regions or the whole genome of interest, and that too in addition to all anti-TB drugs that are used. Currently, it is being recognized and recommended, and it is coming up. You see in the guidelines that are issued by WHO and also by the National TB Elimination Program, Ministry of Health and Welfare, Government of India. So the advantages of whole genome sequencing are it detects genomic variants to phenotypes and guide treatment decisions. It overcomes challenges associated.
Dr. Nikhil, sorry to disturb you in between, but
yes.
There is a request from the attendees that people want you to be slow a bit because content is too much, and your slides are too informative. So people wants to grasp the more. So it would be really appreciable if you can go back from the whole genome sequencing and connect from there and in a slow manner. Thanks, Atan. Yeah.
Sure. Should I start from here?
Yeah, most appreciated. Thank you.
Yeah. Slide visible?
Yeah, clearly.
Okay, thank you. So thank you, everybody. So coming back, whole genome sequencing, also known as full genome sequencing, complete genome sequencing, or entire genome sequencing. This is the process of determining the entirety or nearly the entirety of the DNA sequence of the genome of an organism at a single point in time. So how does it work in MTB? You see resistance in tuberculosis is mainly conferred by nucleotide variations, and what whole genome sequencing does is that it uses high resolution sequencing technology to interrogate the entire mycobacterial genome to identify drug resistance to all the anti-TB drugs that are used. So it can also detect mixed infection, co-infection, and heteroresistance. Keep in mind your GeneXpert will only test RPOB for rifampicin. Your line probe assay will test INHA, CATG, RPOB, and you see Gair is A and EN, the A and PRSL, that will be only for your ofloxacin, amikacin, and ethambutol. It will not test the entire TB genome, and it will not test for susceptibility or resistance to all the drugs that are being used. Right? So how does it work? You see, you have sample collection, then culture enrichment, you have the phase of DNA extraction, pre-sequencing preparation, sequencing, data analysis by the whole genome sequencing test, also called Omega TB test, and then you have the report generation, which is an automated process. Right. So these are the 18 drugs and the genes to which whole genome sequencing will process the sample and test it for Mycobacterium tuberculosis. And you see all these genomes will be tested for the presence of resistance conferring mutations, which are there. They have been displayed here for all of you to see. You have rifampicin, INH, ethambutol, pyrazinamide, streptomycin, capryomycin, kanamycin, amikacin, moxifloxacin, ofloxacin, gatifloxacin, echonamide, linezolid, paraminosalicylate, bedaquiline, clofazimine, delaminide, and phytomenide. So in fact, you see all the drugs which you would consider in a first line regimen, and most of the drugs you would consider while designing an appropriate regimen for a patient of MDR, pre-XDR or XDR TB. Also, you will get resistance and sensitivity patterns beforehand, and this will be of use. So what are the performance metrics you see when it comes to first line drugs? The sensitivity and specificity to INH, rifampicin, ethambutol, and pyrazinamide of whole genome sequencing, the service culture is above 90% for all these 4 drugs. And even for most of your second line anti-TB drugs, the sensitivity and resistance is more than 85% across all the drugs that are being used. So when you compare, you see the agreement between CBNAT and your culture DST, it's just 49%. With LPH, it's just 63%. With whole genome sequencing, it's a whopping 93%. So what are the advantages of whole genome sequencing? How does it resolve an unmet need? How does it mitigate challenges that we have for diagnosis and management of MNXDR TB? It has great potential as a method for rapidly diagnosing DRTB, including MXDR, pre-XDR TB in diverse clinical or reference laboratory settings worldwide. And it overcomes many of the challenges that are associated with conventional phenotypic testing, that is liquid and solid DST, solid and liquid culture, as well as the limitations of other less comprehensive molecular methods such as your gene expert and LPA, by providing rapid detailed sequence information from multiple gene regions or whole genomes of interest. This is what I told you earlier. And then it's being recognized and recommended by the WHO. It is being evaluated for diagnosis of MNXDR TB. It has also found mention in the guidelines for programmatic management of drug-resistant TB in India, as per the guidelines issued by the National TB Elimination Program, Ministry of Health and Family Welfare, Government of India. What are the advantages? It detects genomic sequence variants to predict DRTB and XDRTB phenotypes and to guide treatment decisions. I've told you it overcomes the challenges associated with conventional phenotypic DST, mainly in terms of time. You see, you have discrepancies in your solid or liquid culture for certain RPOB, that is rifampicin or GAIRE, that is quinolone mutants, in different media, as well as the complications of diagnosing pyrazinamide resistance. You see, pyrazinamide is a drug which works only in acidic media, and your liquid and solid culture alkaline media. So there can be a discrepancy between in vitro and in vivo sensitivity. So TB genome sequencing might be considered a more robust reference standard, particularly for certain drug resistance profiles. It also overcomes the limitations of your gene expert and LP and other rapid molecular tests by giving you complete and rapid detailed sequence information. It can also detect heteroresistance, which is a mix of multiple genetic populations in the same clinical sample. It assesses lab cross-contamination and is also useful in the diagnosis of other infectious diseases. And its utility in the public health domain, particularly to the TB control program. It can identify strain lineage and resistance mechanisms for TB surveillance. It can recognize and identify certain genetic strains for the resolution of transmission chains to direct TB control and elimination efforts, not only in countries but also across the globe. However, certain disadvantages are there, like with every test. It is an expensive test. It costs about 15,000 to 18,000 in different parts of the country, but I think Thyrocare, in partnership with Haystack Analysis in Mumbai, is offering it for just 3,000. It's a technically complex test, no doubt. You see, the more advantages a test has, usually the more complex it is. So you see, to integrate it into existing lab workflows will take some time. You need to train your staff, your lab staff, and there are different skill requirements to integrate and utilize this technology in the lab. And also you need to train physicians for clinical interpretation of the sequencing data. There can be some discordance between the detected genotype and the resistance phenotype for some drugs, particularly fluoroquinolones. Now, fluoroquinolones, we all know, are key drugs, particularly in an MDR and XDR TB anti-TB drug regimen. And breakpoint artifacts might lead to misclassification of phenotypes. So a word of caution, ladies and gentlemen, I've said it before, I'll say it again. No lab test is 100% foolproof or 100% foolproof. And phenotypic tests such as your culture and genotypic tests such as your line probe assay, gene expert, or even whole genome sequencing for that matter are no exception to this rule. Everything has some pros and cons ultimately. You have to keep in mind when you're going for rapid molecular methods that all of them for any organism, not just TB alone, they basically rely on the molecular detection of resistance, and that will depend on the presence of the resistance conferring mutation in the genome extracted from the clinical sample. Now, keep in mind that alternate mechanisms of resistance may develop sometimes, and mutations may appear which the test was not designed to detect. Hence, if there is a discrepancy, you have to go back to your phenotypic tests or your culture to identify resistance in case of discrepancy. And again, these tests are slower and subject to contamination, as we have seen. So can whole genome sequencing actually mitigate existing challenges in multi and extensively drug-resistant TB? Let us see. So for that, we need to consider what are the existing challenges. Now, this is a workflow, you see, which will show you. I've already discussed before in my initial slides how it goes. And this is how it goes. Day one, when the TB patient comes to the program, assuming this is a case of pulmonary tuberculosis, you will send a sputum for CBNAT. That is the first step. Though the turnaround time is just 1 hour 45 minutes, it takes at least 4 to 5 days for us to get the report in hand. So there's a delay. If there is some doubt, you send for first line LPA, and then again LPA is not comprehensive enough. Gene expert will identify RIF resistance. LPA, if it is first line, will identify INH and RIF resistance, and second line LPA will identify resistance to ofloxacin, ethambutol, and nicotine. So it's not testing for all the drugs. And then ultimately, you see, by the time you have all this, day 45 to 60, that is 6 to 8 weeks, you will get your culture report. So the mean duration for diagnosis and treatment initiation can be as high as 65 days, even in a new or retreatment case. This is not just DRTB cases we are talking about. We also need to keep in mind, particularly our country, it has high initial resistance to INH, ethambutol, and fluoroquinolones as per the National Drug Resistant TB Survey in 2017. And this resistance is not uniform. You see, it is geographically variable in different parts of the country. Even when you send samples for gene expert and line probe assay, you don't get the results in an instantly. You have to wait often for at least 5 to 7 days in spite of a rapid turnabout time. You see, being a few hours for your gene expert or your line probe assay. And sometimes when the sample is paucibacillary, you say very low, insufficient sampling and all, you have to subject the patient to repeated tests, particularly in paucibacillary samples. Then the patient gets tired submitting the sample again and again. And this is something I have also seen when working with the program for 5 years. There have been issues of non-availability or interrupted supply of diagnostic modalities, particularly the gene expert cartridges. And I've told you before, though we use it as a rule-in test for diagnosis of RIF resistance and to initiate MDR-TB treatment, CBNAT, because it detects only RIF resistance and not INH resistance, it cannot be used to define MDR-TB for program purposes. And there is a lot of time that may be lost in referral and sampling for additional first and second line LPA, and this may cause delay in diagnosis or even inappropriate treatment of MNXDR TB. Though it is used as a rule-in test for diagnosis, the second-line LP or the genotype MTBRSL is still not accepted by WHO to define XDR-TB for surveillance purposes. As cross-resistance between the second-line injectables is incomplete, the second-line LPA or the genotype MTBRSL cannot be used to identify individual drugs to be considered for the initial treatment regimen. It also has to be kept in mind that if resistance is detected in low-risk XDR-TB suspects, a repeat molecular test of culture DST is again advised, and again this will compound the delay. Second-line LPA is less sensitive for extrapulmonary samples due to low bacillary load. Hence, negative tests also need to be correlated, and sometimes you have to repeat the test. I told you about this earlier. Not only shortage of diagnostic cartridges, but there can also be interrupted supply of second-line anti-TB drugs under RNTCP, now NTEP. There have always been, you see, some issues with availability of injectables earlier. And now also from time to time, nearly once a year, we face some issues with availability of drugs like clofazimine, etc., because the raw material has to be exported. So these factors would hamper successful implementation of any rapid diagnosis or even shortened treatment strategy. Let me share a few cases that any of us may come across in day-to-day clinical practice, which will explain what the challenges in MNXDR TB are. What if your patient is unable to give repeated samples and is unable to travel frequently for lab reports? What is the solution? You don't try and send the patient for the test. Instead, you try and bring the test to the patient by a home visit, by active case finding. You don't wait for the patient to come to the TB clinic with complaints of cough or expectoration or weight loss for 2 weeks, etc. You go to the patient's house. You go to the locality. You ask how many patients are suffering from fever, cough, weight loss, and then zero in on those cases and test their samples, what we call active case finding, instead of the passive case finding we are doing currently. And then digitization, where you get the lab reports in hand. And I'm thankful the program has implemented this partly through Nikshay, which is being used for TB notification. It also has scope for digitization of patient reports as well. I told you, if your sample is possibacillary, you will get inconclusive reports on CB-NAT or LPA. Sometimes the patient cannot be classified as a case of MXDR TB due to test-related discrepancies. Sometimes the patient's condition will deteriorate and you cannot wait for a phenotypic culture or DST, which will take anywhere from 6 to 8 weeks. So what will you do? So again, my friends, you should offer a patient as well as physician friendly test by considering at least TB whole genome sequencing at the outset, which will give you see 18 drug resistance or sensitivity profile within a period of 15 to 18 days. Again, what if your patient has extrapulmonary TB, TB outside the lungs, or for that matter, multifocal TB in the lungs, or at more than 2 or 3 sites? Where will you take the sample from? If the patient has already taken anti-TB drugs previously, therefore being at risk for drug resistance, is receiving an appropriate but failing anti-TB drug regimen, is facing diagnostic delays or issues with gene expert or LPA, which I have discussed earlier, is suspected to harbor cross-resistance or heteroresistance in the MTB in his or her clinical sample. Again, TB whole genome sequencing at the outset can overcome many of these problems. Again, the gold standard for treatment in any patient of MNXDR TB, for that matter, even a patient of drug-resistant TB, is an individualized anti-TB drug regimen where you will give the patient drugs, a regimen of drugs, or you will design an anti-TB drug regimen, choosing only those drugs that the patient is sensitive to. So you see, with gene expert or line probe assay, it may not be possible because there is incomplete information or unavailability of DST to a lot of other anti-TB drugs also. They are useful for designing basic standardized regimens, but not for individualized regimens. What if your patient is on an appropriate regimen, is unable to tolerate certain anti-TB drugs which have been started, and it's required to change or substitute with other anti-TB drugs? Now, this will put you, the treating physician, in a dilemma because you don't have information on which drugs may be effective. You don't have a complete DST profile to anti-TB drugs. Whole genome sequencing will give you this information at the outset. So this is a solution that will help. And what should be the diagnostic pathway, and where can whole genome sequencing fit in the TB diagnostic pathway? You see, I've given it here, and I thank Haystack Analysis for providing me this slide also. You see, you can do it at outset, sputum smear, CBN at day 1, then you get RIF resistance on day 2, and then by the time you see at day 14 or 15, you will get a whole genome sequencing report in hand. So it will cut short the time that is currently anywhere from 45 to 60 days minimum for diagnosis of drug-resistant TB to just 14 days. Similarly, when you are having a retreatment case, Similarly, when you are having a retreatment case, when your LPA fails, again, this will help you. Drastically, you will get a report in hand at the outset. And then in critical retreatment, that is to say a patient who's failing an appropriate regimen or HIV comorbid case, you can get whole genome sequencing reports much faster, provided the program prioritizes it. So what are my views, if you ask me personally, friends, for any rapid intervention or any other intervention that shortens the time for diagnosis and treatment in TB, including MNXTR TB, to be effective to reduce the TB burden by morbidity and mortality reduction or cutting down the chains of transmission, which we know are the aims of TB control and now TB elimination globally. We need to look beyond conventional case detection, cure or conversion rates. We also need to look at relapse rates for up to 5 years after treatment completion, particularly in cases of MNXDR TB. Friends, unless we tackle key issues related to DRTB, creation, proliferation, spread, case detection by active case finding, treatment and prevention, just shortening the time towards your diagnosis or giving them a shortened rapid regimen, you see, alone, these two will not help. And neither will, cutting down the time frame for NTB strategy by 5 years from 2030 to 2025. No doubt it is a well thought of and ambitious target. But simply by doing this, we will not be able to catch up with the large number of MNXDR TB cases we as a nation produce year after year. And though I've worked with the program, it's a good program. I don't wish to criticize too much, but I am also aware that there have been certain policies all the while. You see, historically fallacious policies for the last 15 years also have contributed to the MNXTR TB burden through diagnostic delays, which I have discussed, through treatment failures, which I have discussed, lack of access to quality assured services. Not that government was not providing, government was providing the best diagnostic and treatment services, but many patients and physicians also were not aware where these are. So, there was limited access to these facilities. And this often from time to time, I have seen this compelled patients to give up on the public healthcare system and go to seek inappropriate management, be it diagnosis or treatment elsewhere. DOT, that is directly observed TB treatment, needs to be retired in order for a more robust and successful strategy to emerge. Thankfully, this process has begun. I'm very thankful that it has been corrected. We need to know that any delay in diagnosis means a delay in treatment, and any delay in treatment means time, productivity, and life years, and in some cases, even the life is lost. Time lost in even one case means more morbidity, mortality, suffering, spread of infection from patient to family, caregivers, close contacts, and society. So that one case will amplify, you see, so many more. Any delay is a burden on patient, family, and society, the healthcare system, and also on the economy due to productivity loss. So we must strive to establish prompt and definitive diagnosis of DRTB, whether it is multi or extensive or pre-XDR or XXDR TB at the onset itself. We need to keep in mind that patients with drug resistance or multi or extensively drug resistant TB, they often exhibit variable patterns of sensitivity and resistance to the commonly used anti-TB drugs. And therefore, what whole genome sequencing will do, it will give you upfront knowledge of which drugs are likely to be effective prior to starting therapy or even during therapy in case you need to change the regimen. And this will help you provide effective treatment right from the beginning because you know what you are dealing with, what is likely to be effective and what is not likely to be effective. So you see, the need is that TB patients need timely diagnosis and care. The hurdle is that current culture and molecular tests will give you limited information, or if they give you complete information, will take at least 6 to 8 weeks. The advantage of whole genome sequencing is that it can be a single point of care test at the outset to identify all your diagnostic information and DST profile to Mycobacterium tuberculosis. It just needs to gain acceptance and is already slowly but steadily gaining acceptance by the authorities. Whole genome sequencing is the new kid on the block, which may prove to be one day the one-stop upfront test for diagnosis of drug resistance, multi or extensively drug resistant TB, with the potential to steer patients along the right treatment path early. And finally, I'll end by showing the photographs of these two famous gentlemen. We all know them as Dr. Louis Pasteur and Dr. Robert Koch, who would be happy that it's about time we have a good rapid test which gives us complete information. These are a few references, and with this I conclude. Once again, I'd like to thank Haystack Analysis and Thyrocare for inviting me here. And I thank you, Dr. Chaitali, for moderating this session and all our participants for being with us. Thank you.
So thank you, Dr. Nikhil. Thanks a ton for such an informative session, which is covering almost all the aspects of molecular and phenotypic and genotypic methods, along with given a brief introduction about the WGS, its technology involved, the targets and the difference between the target sequencing and whole genome sequencing, regimens, patients' treatments, and a lot more. And there are few questions, so I would like to take them one by one. So there is a first question which says about CFID has launched XDR cartridge last month only, getting results in a runtime of 90 minutes in that from GeneXpert. So how WGS will help because it takes 12 days for results? May you please make me understand how WGS is more effective than existing technology that is GeneXpert? So would you like to take this question or should I take it, Dr. Nikhil?
Yeah, I think we both can take it, Dr. Chaitali, since it's a question that deals with the microbiological aspects also. I'm sure you can deal with it.
Yeah.
But from my end, I think I'll just clarify. You see, the CFID cartridge will not give you resistance and sensitivity profile to 18 anti-TB drugs, unlike whole genome sequencing. And that I told you, I've discussed in my talk. It is important, particularly when dealing with a drug-resistant case, whether it is MDR, XDR, etc., to have this information on hand at the outset. And this is, you see, of paramount importance because if you need to, you know what drugs to begin with. Right now, what we are doing, you see, we are getting, diagnosis of MDR and XDR TB by GeneXpert LPA, but we don't know which drugs are sensitive apart from the key drugs that have been tested. You don't know what other drugs to give. You are just designing a standardized regimen, anti-TB drug regimen, because the patient is classified as MNXDR TB. But the gold standard is not that. The gold standard is an individualized regimen. You start with 6 to 7 drugs to which you know the isolate in the patient sample is sensitive. So there you know you are going to give the patient a standard, the best possible standard of care. So I think Dr. Chaitali, you can add to that.
Yeah. So this question is from Mohan Agarwal. So I'm totally agree with Dr. Nikhil what he has said about, but going deep down to this GeneXpert MTB XDR cartridge, that is a 10-color cartridge which has launched in just last week. So I would like to give a clear insight about this cartridge is first of all costing, which has been not disclosed yet. Another thing which is very important technically, that this cartridge gives information about isoniazide, ethanamide, fluoroquinolones, and aminoglycosides. Now, in aminoglycosides and fluoroquinolones is same as a group of antibiotics which comes under line probasis, and in fluoroquinolones, gyrase A and gyrase B. These are the gene targets which has been, targeted, and the codon region which has covered is 87 to 95 for gyrase A and for 531 to 544 for gyrase B, which is again the similarity of LPA. So you should understand that if any other mutation, if this is a present, beside these codons, it may missed in this. Also for second line injectables or aminoglycoside, where RRS and EIS promoter region have been targeted in XDR TB cartridge. Also, this is, as Dr. Nikhil has rightly said, that it is not giving you the information about the 80-90 TB drugs. Beside that, it is having in the system, they had given the distinguished features of a phenotypic DST and sequencing sensitivity and specificity. So for kanamycin and capreomycin, it is having sensitivity of 87 and 84% respectively. Whereas for etinamide, it is having 67% sensitive as compared to phenotypic DST, not with sequencing, but phenotypic DST. So all these things matters when any patient's decision or any patient's treatment needs to be decided based on such kind of a test. So Dr. Mohan, hope we have answered your query satisfactorily. Moving ahead to second question, which is by Ganesham Mishi, and it says about, are silent mutation a problem with WGS? So would like to provide your email address, Ganshankar, because there are publications which says that WGS is a technology which can be used to resolve the discrepancies which is happening in phenotypic DST due to silent mutations. So Dr. Nikhil, do you want to add something over the silent mutation part?
I think, yes, you see, it's a very important question. And even we have seen that when we send patients for liquid DST, a small proportion, very small proportion, but significant nonetheless, they also have this problem of silent mutations in the sample. And this can really confound, because you are relying as the clinician on the DST report of liquid DST to give you information on the drugs to start. And like I said, there can always be a difference between in vitro and in vivo sensitivity of the drugs. So you are stuck there. And if in certain patients, silent mutations are also present, then you're stuck still further, because you think you are giving the right regimen. You think all the drugs that I've tested for, to be sensitive in the liquid DST will also work on the patient, but then it is not actually so.
So yeah, so absolutely agree with you, Dr. Nikhil. Now, would like to move to next question, that is from Dr. Paj Shah. What all samples that can be processed in WGS? So all sputum, that is all pulmonary and extrapulmonary samples can be processed on WGS. Only we need to check about the smear status, because if smear status is positive, definitely you'll get the result in a short period of time. That is a 12 days of a WGS stat, which we have mentioned. But if it is a smear status negative, as we all know that extrapulmonary samples are having less load of a bacteria, and then it becomes tricky to extract the DNA and good quality of DNA, because WGS requires good quality of DNA. So if it's a good sample, good quality sample, then definitely all types of samples can be catered for WGS. Next question is from Dr. Suresh Kumar Mishra. He wants to know, sir, and I would like to ask you to take this question. So his question is, for bone, should we collect the bone marrow fluid for diagnosis of TB test? So over to you, Dr. Nikhil, for this.
Yeah, so you see, when it comes to bone TB or osteoarticular TB affecting the bones and joints, what we commonly see in adults and in pediatric patients also, the most commonly affected sites are the spine, all right, tuberculosis of the spine, tuberculosis of the hip, tuberculosis of the knee, and then of certain other joints. So bone marrow sampling is not actually recommended for TB diagnosis. What we collect, you see, suppose we have a case of TB spine, what we usually collect is the biopsy of the sample there, the whole, you see the whole specimen that is extracted during surgery, or for that matter, most of these patients also have associated psoas abscesses. So you can just drain some of the fluid from those psoas abscesses and send that for your testing, for culture DST, whatever, or for genome sequencing, whatever it may be. So such samples are preferred rather than bone marrow. Another thing is that when you are doing bone marrow sampling, there is always a risk of transmitting infection. Most of the time in osteoarticular TB, the bones and joints have become weak, and to do sampling directly from the bone marrow may not be a very good thing. Bone marrow sampling is very useful when you have TB in association with some other disease, hematogenous, for example, lymphoma or Hodgkin's disease. There, yes, you need to do bone marrow sampling, but not primarily for TB diagnosis. It is primarily for the diagnosis of the coexisting disease. I'm not sure if samples from osteoarticular sites such as psoas abscesses or direct sampling from bones or synovial fluid in some cases has been evaluated with whole genome sequencing, but I think more data on this will come up in some time.
Thank you, sir. So there are no more questions here in the chat box, but yes, it's chat box is saying very good presentation, and thank you, Dr. Nikhil, for such a lovely presentation again. And thank you, Team Haystack and Thyrocare for arranging the session. So with this, I'll conclude the session. And yes, there is one question in this here. How to collect sample from patients? What's the medium of sample transport? So sample, I'll take this question. Sample can be transported into the sterile container. So Dr. Nikhil, lots of comments for your nice presentation, and I'm just conveying to you as a moderator. And with this, I'm concluding the session here. Thanks, Atan.
Thank you.
Thank you, everyone.